Transgenesis is a term that denotes an animal with DNA that has been artificially introduced. Essentially, there are 4 methods used to influence an embryo to become transgenic, which include recombinant DNA, or artificially produced DNA, genetic cloning, analysis of gene expression, or the process by which a gene gives rise to a protein, and genomic mapping. However, the most common is recombinant DNA, which is introduced to the sample animal via microinjection, retroviral-mediated gene transfer, and embryonic stem cell-mediated gene transfer, also known as ES cells. Scientists can inject DNA into the embryo of an organism in order to knock out a set of genes known as homeotic genes, which govern body patterning throughout development. In order to initiate the process of transgenesis, an individual must identify the gene that they must work with. For instance, if the gene known for causing lactose intolerance in mice is identified, then it can be experimented with in order to see how it affects offspring. Firstly, they must isolate the original ES cells with the preferred gene from the blastocyst. Next, they add a mutated copy of the specific gene in addition with the drug-resistant gene marker, which kicks out the natural gene and takes its spot in the ES cell for an unknown reason, a process known as homologous recombination. Following this, cells that haven't accepted the mutated gene will not have the drug resistance factor. So the scientist will usually add a drug that will kill the remaining cells, leaving them with only the inactive and mutated copy of the gene. This tampered blastocyst is then transplanted into a pseudo-pregnant female in order to begin the next generation of chimeras, a single organism with different cells from more than one zygote. The resulting mice will then have a copy of the inactive gene from the father and an active copy of the gene from the mother. Therefore, with more breeding, they will eventually have mice that have only the inactive genes. Cows can be modified to produce insulin in their milk or proteins that kill bacteria that cause mastitis. In labs, techniques to perform this can be used to obtain information on gene regulation and function for human diseases and recombinant pharmaceuticals for human therapy. In addition, new ideas can be formulated for xenotransplantation, such as engineering a pig organ that can be inserted into a human body and function properly. Transgenesis has already been performed on animals such as mice, rats, rabbits, pigs, cows, and sheep. Therefore, it is beginning to raise questions regarding ethics. For instance, the implementation of universal protocols, the welfare of other life forms, the focus of in vitro transgenic methods rather than, or before, using live animals to alleviate animal suffering, the change of the direction of evolution, and patents. Regardless, it could be a pathway to a new future for our medical field.